Widal test for the diagnosis of typhoid fever. Widal test is a serological method to diagnose enteric or typhoid fever that is caused by Salmonella typhi and Salmonella paratyphi A, B, and C strains. So you know the typhoid is a worldwide illness transmitted through food and water. So once the individual ate food or drank water, once it entered into the individual intestinal wall, this bacteria will absorb, re-enter or perforate into the body blood, and it is phagocytized by the macrophages. So upon the activation of macrophages, there will be a production of antibodies through the B lymphocytes. This is what happens after the individual is infected with the Salmonella species. So after activation of macrophages, there will be a production of antibodies in the infected individual blood. So this Salmonella bacteria is a gram-negative short bacillus bacteria, and it is motile in nature due to its peritrichous flagella. Flagella is a hair-like structure which is used to swim or move from one place to another. So the term enteric fever, it comes from the genus Salmonella, belongs to the Enterobacteriaceae family. Enterobacteriaceae family, causing the diarrhea also. Salmonella have three types of antigens. One is flagella, somatic, and surface antigen. Flagella antigen is always called as H antigen. Somatic antigen called as O antigen. So this is present in the gram-negative Salmonella bacterial cell wall. Containing lipopolysaccharides. Surface antigen present in the outside of the bacterial surface. So it's called VI antigen. VI that indicates the virulence antigen. Virulence means ability to cause the diseases. So this antigen is responsible for the infection transferring from one individual to other individual. So what's the principle involved in the Widal test? So Widal test is working based on the antigen-antibody reactions. So this case, agglutination reaction takes place due to the antibodies present in the patient blood and the antigens specifically prepared from the Salmonella species provided by the Widal test kit. So there will be an antigen-antibody reaction showing the agglutination. It's caused by the antigen present in the kit plus the antibodies present in the patient serum or blood. That is making the agglutination reaction, which is able to observe through our normal naked eye. So this is the principle involved in the Widal test, that is antigen-antibody reactions causing agglutination. So this test is described by Fernand Widal in 1896. So it's a very old test, but still it is there in the clinical laboratory as our physicians are recommending this test for the diagnosis of typhoid fever. You know, old is gold. So what is the components of Widal test kit? So Widal test kit consists of the antigen suspensions of Salmonella species. So totally 8 antigens are present in the Widal test kits. So including Salmonella species O and H antigen and Salmonella paratyphi of A, B, C strains having O and H antigen. So moreover, it has negative control and positive control. Negative control contains no antibodies against the specific bacteria, and positive control contains standardized gold antiserum with polyspecific antibodies. So this is specific reactivity towards Salmonella typhi and Salmonella paratyphi O and H antigens. So it's useful in validation and performance of the Widal reagents. So this is the kit. Contains 4 plus 4, 8 antigens, then negative control and positive control. So what is the procedure involved in the Widal test? It's a simple, just adding 1 drop of blood in the each circle, followed by adding 1 drop of the reagents in the test kit of all antigens. So blood plus reagents, then we need to mix and spread with the stick provided in the kit. After mixing, we may rotate the slide for 1 minute, then go for observation of agglutination. So observation of and results. So there is no agglutination reactions. It's a negative. There is no typhoid infection in the individual blood. If there is agglutination present, if agglutination present in the O antigen, it indicates that it's a recent infection, just is an acute infection. Recently, an individual infected with the Salmonella typhi. If the infection or agglutination showing in the H antigen, that means that it's an old infection, it's a chronic, the infection is prolonged. If the test showing agglutination in the para-typhi O and H antigen, it means that the individual may serve as a carrier and he can able to infect the other. So other is the tube agglutination test. So the Widal test may perform in the slide or tubes. So previously we have seen the slide. This one is the tube agglutination. So these are the tubes having the different kind of dilutions, 1:20, 1:40 accordingly. Right? In the tubes we are mixing the Salmonella antigens plus patient blood. Right? After mixing, we may wait for some time for showing the agglutination reactions. So normally the results were reported based on the titer. So titer is the concentration, highest dilution where agglutination is seen. So in the tubes, the fourth tube showing the agglutination positive results. Right? Later on, this higher concentration also showing the positive results. You can see negative, 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 and up to 1:80. So 1:60 showing the positive results. So the titer starts from 1:160. So this is the tube agglutination test. So what is the limitation of Widal test? The only limitation is the cross agglutinating antibodies. So the presence of cross agglutinating antibody, the results may less sensitive and there is an interpretation is very difficult. So this cross agglutinating antibody is present in the patient blood due to some past infections. So previous infection related antibodies may cause negative results or false positive results. False positive results. So this false positive results also due to the previous immunization with Salmonella antigen or other non-typhoid Salmonella or the ELISA test kit is substandard due to the substandard production of ELISA test kit, or it may be with the malaria infection or with the other Enterobacteriaceae having the same surface antigen of lipopolysaccharides. That means the negative bacteria, gram-negative bacteria genus from Enterobacteriaceae family may have a same antigen. It may also provide the cross-agglutinating antibodies that may interrupt the results. So this is the limitation of Widal test. However, the Widal test is considered as a gold standard test still by our physicians due to the low cost and less time consuming. So these are the other tests available in the market for diagnosis of the typhoid fever. That includes typhoid test that is based on the immunoglobulin G and immunoglobulin M with the chromatography. can be done within 15 minutes. Then Tubex TF test within 10 minutes we can perform. ELISA and ECA electrochemiluminescent immunoassay. So these tests are based on the antigen antibody reactions only. All the tests are based on the antigen antibody reactions only, except the PCR polymerase chain reactions. Thank you.